Fine. So hello, hi everyone. So welcome to the second day of this particular workshop. Okay, and in today's lecture, as I had announced, that we'll be trying to cover and look at the data preprocessing, normalization, and few other aspects used for the process of gene expression data analysis, right? And the same process that we use for the gene expression data analysis are also used upon either methylation, microRNAs, and other data sets, because the data science remains the same, right? The complete flowchart remains the same. It's only the different techniques will be changing. And if I ask you like the techniques that will be changing will depend upon what? The simple answer is that based upon the sample preparation, the process by which the samples were generated in the lab. Okay, so based upon that only the different techniques changes, right? So if we have to start for today lecture, so see, like I'm going to give you idea about the gene expression data analysis. Right. So see, like yesterday we had already covered basically that how do gene expression data look like, right? So what did we see basically is that that on the rows we have the different samples, and over the columns we have the different genes, right? Okay. And now what we need to have one more thing, that is the condition. We need to also have the condition vector with us, right? Condition vector, which will help you to understand that the given gene belongs to the given sample, which might be patient, which might be anything, belongs to which particular biological condition, basically, right? So, like we have the sample here, and suppose here it is written BC. BC means here breast cancer. It means what? That this particular sample one belongs to the category of breast cancer. The sample M, suppose it is written here that LC, lung cancer. It means what? This particular patient sample M belongs to the lung cancer condition, right? So ideally the data set should look like this. Okay. And now if I'll ask you a question like what is the end goal of us, right? What is the end goal? Then we know that we're interested to find out the set of differentially expressed genes, right? Differentially expressed genes, right? This is our end goal basically. Like we want to find out the set of the DEGs, right? The set of the DEG, differentially expressed genes, right? And we know like with genes we call it DEG. Basically simple, like any gene which is expressing in a different manner, and the difference should be statistically significant in condition A as compared to the condition B, right? So that is the meaning of differentially expressed genes basically. Just give me a minute, okay? Let me share the link with Dr. Anjali, otherwise it will be a problem for her. Fine, right? So the set of the genes which are expressing differently in two conditions, and the difference should be statistically significant as well. So now see that what we did see last class, that there are two genes, not two genes, one gene, but two conditions, right? And if you're looking at the box plot, then here we see that there is no such significant difference here, right? There is no such significant difference, right? So this is condition like situation one, situation A. Now another condition is that, so this is C1, this is C2, condition one, condition two. C1, C2. Now here we clearly see that the median expression value of the gene in condition 2 is higher as compared to condition 1, right? And if you are talking about C2 upon C1, okay, C2 upon C1. So what do we mean by C2 here? C2 is the expression value of the gene in condition 2. And C1 means expression value of the given gene in condition 1, basically. And the mean, we're talking about here the mean, right? Because we know it is impossible to observe each of the value, and definitely we need to have some metric, the metric which can help us. Okay, we need to have some metric, the metric which will help you to compare the data in the two different conditions, basically, right? Okay, so, and the condition 3 basically here, it looks like this. See, now it is basically C1, it is C2. Now again C2 upon C1, if you're going to compare the ratio of the medians, okay, median values, then we'll find that here it is having the lower value, okay, lower value. So based upon this, we have total 3 conditions, okay. One condition is that, one possibility is that no significant difference. Situation 2 is that upregulated. Okay. And the situation 3 is basically downregulated. Right? No difference, it means that the expression value of the given gene in the two different conditions is the same. Upregulated means the expression value of the gene in condition 1 is greater as compared to the condition B. And downregulated means expression value of the gene in condition 1 is lower as compared to the expression value in the condition 2. Right? Now we know. So let me ask this question to you guys here. So, Yanjusha, what do you think? Like the maximum number of the genes should be up, down, or no significant difference? So what do you think, Yanjusha? That majority of the genes हमारे क्या होने चाहिए? Different होंगे कि नहीं होंगे?
नहीं होने चाहिए.
नहीं होने चाहिए. Okay, fine. So Hariom said that नहीं होने चाहिए. Okay. And Hariom, what do you think? So नहीं होने चाहिए?
सर क्योंकि मतलब सभी मतलब डिफरेंट कंडीशन में ऐसा तो मतलब अगर मैं डिजीज की बात करूं, एक डिजीज हो एक नॉर्मल हो, तो ऐसा तो होगा नहीं कि सभी जीन डिजीज के रिस्पॉन्सिबल हो. कुछ स्पेसिफाइड जीन ही रिस्पॉन्सिबल होंगे, इसके मैक्सिमम नहीं होने चाहिए.
Good. So that is absolutely the right answer. And another logic behind that is that we also know that there are a number of genes as the housekeeping genes, right? So the genes housekeeping means that they are very highly important for the normal functionality of a human, right? And these genes you will find that will have no change in the two in any of the conditions. Basically, they will always be present, right? They will always express. Okay, so these genes are known as the housekeeping genes, basically, right? So there are the different reasons behind, like why do we find very low number of the genes are differentially expressed? Okay, majority of the genes, why do not they express with significant difference and all, right? So now, if you're going to now look or keep it into consideration these facts, then we have calculated one factor which is known as the fold change. Basically, right? Fold change. What do you mean by fold change? Simple. Fold means times, right? The mean expression value of the gene in condition 1 divided by mean expression value of the gene in the condition 2. And it is the gene. Okay. So the mean expression value of a gene divided by mean expression value of the gene in condition 2. This is named as fold change. Okay. Now, if you're going to take the log of this, then it will become what? Log of fold change, right? So now what we do basically is that usually we, at the time of pre-processing the data, or making the data ready, we normalize the data at that time only. Okay, we normalize the data at that time only. So when you have already normalized the data, then this log fold change, it becomes log condition 1 gene minus log C2 gene. Why? Because we know that log of A upon B is equal to log A minus log B, right? And then log A dot B is equal to log A plus log B. They are the properties of log, right? So we know this thing. So using property number one, we subtract like the means are basically get subtracted at the log scale, right? So this is how log fold change is calculated. If the data is already log transformed, then you will subtract the values, the means. Okay. If the data was not log transformed, then you will take the log of this, log of the means basically. And we take the log on the base scale of 2. Why? Very simple, because on the base scale of 2, it means that double. Because we know log of base A and A. Whenever the base and the number are the same, it becomes 1. It means what? If a gene is 2 times, the ratio is 2 basically, and the base is also 2, it will become 1. Okay. And suppose log 2 0.5. Okay. It means gene is down regulated. Then it will become what? Negative. And I explained you it in last class. Third case, if it is greater than 1, it will become what? Positive. It means what? So we are going to have a symmetric diagram basically. So 1. So when the expression value is 1 basically, so log 2 of 1 is equal to 0. Okay. Log any base of number 1 is always 0 basically. So it will become 0. Okay. So 1. So value 1, it will become 0. And then it will be positive. It will be your negative. Okay. Then we need to find basically that this is the log fold change. Here we have only taken into account the gene is either differentially expressed or not, right? But now we also need to take into account the sample size, that the results that we are calling, the genes that we are calling as differentially expressed, are they really significant different or not? Okay. We need to claim that the sample size that is present there, are these enough in order to claim a gene to be differentially expressed or not? Okay. And that is being given by, we know this concept. Can anybody tell me how do you claim or basically declare any value as significantly different or not significantly different? What is that particular statistical term named as? Anybody?
P-value.
P-value. Good. Okay. So p-value we make use of in order to declare a gene as significantly different or not. So Hariom, can you now explain p-value?
सर ये अभी बेसिकली एक थ्रेशोल्ड वैल्यू है, मींस अगर हमारा अल्फा इस वैल्यू से ज्यादा आएगा तो हम हमारा जो हाइपोथेसिस है वो रिजेक्ट हो जाएगा, नहीं तो फिर हम उसे एक्सेप्ट कर लेते हैं।
चलो आपने थोड़ा उल्टा बोल दिया, बाकी सब ठीक है, ठीक है। सो नॉर्मली ओके फाइन, सो लेट मी एक्सप्लेन यू नो द कॉन्सेप्ट लिटिल बिट। सी, so this is basically how the density curve of normal distribution looks like, right? We know this thing. And now here, this is the probability basically. The area under the curve is probability. This becomes 1. If you're going to summate the area under the curve, it will always become 1, right? Now, and there is some term named as the false positive rate. False positive. So I will explain you. So let me explain this. Because in the hands-on, I will just show you all the thing using code. That is not a big problem. But you need to know how to interpret the results. For that reason, I'm teaching you all the fundamentals. Okay. So see, what happens basically is that there are two possibilities. One is H naught, other is H1. H naught is one, what? H naught is one null hypothesis. And H1 is what? Alternate hypothesis. Okay. Now, there are there are multiple possibilities. Out of those, we are using here two possibilities. See, H naught is true. H naught is false. Reject H naught. Fail to reject H naught. P-value, P-value. See, in the case number 1, we have written probability of rejecting H naught. or rejecting the null hypothesis, given that null hypothesis was true. It means what? Null hypothesis was true. In reality, like it was true, but still you are rejecting the null hypothesis. So it is termed as, there are two type of error, type 1 error and type 2 error basically. So can anybody tell me, like, this is type 1 error, type 2 error? Rejecting H naught given H naught is true. Anybody would like to try?
Sir, I guess a type 2 error.
Okay, so you're saying type 2 error. The answer is wrong. So it is basically type 1 error basically. Okay. And the other possibility is that H naught is false. So in reality, like in the true case scenario, H naught is false. Okay, but you are failing to reject H naught. You are not accepting, but even like you do not have the sufficient evidence even to reject that null hypothesis. Okay, so it becomes your type 2 error. Okay, guys. So these are the two different possibilities of errors, type 1 error and type 2 error. In any of this statistical testing, whenever we perform any hypothesis testing, basically, it is impossible. It is impossible to not to make type 1 error, basically impossible. Okay, it is always that you'll make some type 1 error. Okay, and we always take this thing into account or assume this thing in the beginning of the experiment, and we assume that we'll make some false positives. It is also named as false positive rate, false positive. Okay, and it is named as false negative basically. Okay, guys, so it is always fixed that you will make some type 1 error. And for that reason, we always assume in the beginning of the experiment that we will make some error. Okay, we will make some error. So if you're going to make some error, okay, if you will make some error, so what we do basically is that we assume that error percentage and we declare that that with alpha, that we declare with the alpha that we are going to make either 5% error or 0.01%, 1% or 0.1%. Sorry, 1% basically. This depends upon the researcher in the beginning of the experiment. Okay, guys. So we always declare that we are assuming that 5% is going to be a type 1 error, false positive rate, or 1% or 10% basically, right? And this becomes your alpha value. It is also p value only. P means probability. It is also p value. It is also probability value. And what is the probability? 5%. So if somebody is going to ask you, like, what do you mean by type 1 error? Simply say that null hypothesis is true, but still we are rejecting the H naught. That is meaning of false positive. And type 1 error is named, also known as the false positive basically, right? So now what we do basically is that we are given the data and everything. Okay, and then using some hypothesis testing, like yesterday in the class, somebody said me that, and yes, that the data is normally distributed or data is parametric. So we can make use of ANOVA and parametric test, right? So definitely like we are going to make the use of some parametric test. Okay, and one of the parametric test is basically your t-test. Right now we are having only two cases. Okay, see, we have only two case scenario, A and B. Okay, A is what? Condition 1, B is what? Condition B. Okay, so we can make use of t-test. What is a t-test? Assume basically. Can anybody tell me here that what are the assumptions of t-test, guys? Hi, Gurleen, are you there? Hello. Anybody would like to try and tell me, like, what are the assumptions of t-test? Normal distribution, yes. So this is somewhere the assumption of t-test basically. Normally, like t-test in itself is a distribution, okay, which looks similar to t or like normal distribution, okay, but it assumes that the data will follow a normal distribution. Not the data, but there is something like I will explain you that thing to you later sometime if you're interested. That what are the assumptions of t-test in detail, okay. But yes, data should be normally distributed ideally. Okay. So fine. So yes. So now what is the task? H naught is what? Mu A is equal to mu B. That is the mean expression value of the gene in condition A is equal to mean expression value of the gene in condition B. Alternate will be what? It is not equal. Alternate will be, it will not be equal, right? Now what you will do, you will calculate the t statistic. You will calculate t statistic, and corresponding to the t statistic here in the distribution, like on the x-axis, these are the values. On the y-axis, you have the density, right? So your t statistic will fall somewhere here basically. And we have assumed what? The alpha is 5%. So 5% error will be what? Somewhere here. So suppose that your t statistic is here. You got your t statistic here basically, right? Now, and you have calculated t statistic, and this is your threshold t statistic basically. Threshold. The t statistic corresponding to the 5% error rate. The t statistic corresponding to the 5% error rate basically. Now, and this is the threshold. Okay, and you have calculated. Now you have, and corresponding to the t statistic, you will get the p value. You will get the p value. And what we are saying that 5% error rate, it will be where? Like it will be here, somewhere here. And the t statistic that you have calculated. Now suppose that there are only two scenarios: calculated t statistic and hypothesized t statistic, or the threshold. If you calculated one is greater than t hypothetical, then it will lie where? In the shaded region. We know this thing, right? It will lie in the shaded region here. It will lie somewhere here, right? And if it is going to be smaller than that, so it will lie somewhere here basically. Now, if my calculated t statistic is greater than this, so my p value is going to, like, probability is going to be less than the alpha. Okay, probability is going to be less than alpha basically. Okay, and the p value is this only, that what is the chance that I am getting the results by chance? What is the probability that I am getting the results by chance, right? So the p value is very, very less. The p value is very, very less, right? And error rate is less than 0.05. So what we do basically is that we say that the results are not by chance. The probability value is very, very less, even less than 5% error rate. Okay, because you're declaring that the results are not being obtained by mistake. Okay, so if now here the decision making comes, t statistic calculated is greater than theoretical t statistic, then you reject h naught. So if you are rejecting h naught, then what do we say? That mu A is not equal to mu B, or we are basically failing to reject the alternate hypothesis, right? So you're failing to reject the alternate hypothesis, and you will say that alternate hypothesis is true. That is, the gene is significantly different in the given scenario. Okay? The results are not being obtained just by chance, because the results being obtained by chance, the error of getting the results by chance is very, very less, right? So p value is that only. What is the probability that the results that you are getting are just by chance? What is the error rate, right? So you are saying that the error rate is very, very less. Okay, and for that reason, you are now going to failing to reject the h naught. So HA basically alternate hypothesis. And so your alternate hypothesis will be considered true here in this case, right? So this is how the complete decision making works. Okay, now see here. Now we know that the p value are, again, these are the values, and p probability values, so it will always be between 0 and 1. And it goes even in the order of 1e minus 30, 1e minus 150, 1e minus 300, and all. 1e minus 300, it means 1 into 10 to the power minus 300. Now it is impossible, basically, like you can't even think of, like, or you can't even imagine like how this value is going to look. How much small value is this? So for that reason, in order to make the numbers comparable, or at the level where you can think of and visualize the numbers, what we do basically is that we take the negative log of p values. Okay, we take the negative log of the p values, guys, basically. Okay, so now if you are taking the negative log of, suppose, minus 10. So that I will explain you later, basically, Priyanka. Okay. So now, so minus sign, I told you the property of log. So log 1 plus log 10 minus 10. Log 1 I told you 0 plus, and log a to the power b, it becomes log b log a. So it will become minus 10 log 10 minus 10 log 10. Okay, and it will become some value because the base is 2 here, base is not 10. So it will be some value, minus positive. So it will be 10 log 2 10. It will not be 1 because base is different here. Okay, guys. So this is the result that you will get. Okay. And now it is definitely comparable, right? Now, one thing happened basically here. That is, there is a problem named as multiple hypothesis correction, multiple hypothesis testing. What is this? See, we have a number of genes. We have high number of genes, right? We have approximately 20,000 genes, right? And we are performing the hypothesis testing here. How many times? If 20,000 genes are there, so definitely we will perform the t-test for 20,000 times, right? And for once, the error rate, the alpha rate is what? 0.05. So if you will go and read basically in detail about the statistics, then you will find that the error propagates in case of multiple hypothesis testing. Okay. And it says that if you're performing the hypothesis simultaneously and continuously for number of times, then it propagates. Okay. So the error will basically increase. Okay. And the chance of making the error in the case of 20,000 tests, it will not remain 0.05, and it has increased basically to some other value, right? So in order to correct that particular problem, in order to correct or take into account the problem of multiple hypothesis correction into account, we correct the multiple, we correct the p-values. Okay, because now the error rate is propagated, and as a result of which the p-values that you are getting, these are not the true ones because error has propagated. So you cannot use the original alpha basically, the original fold type 1 error rate you should not use. Okay, so when you should not use the true alpha rate, basically alpha value, so what value should we use? And for that reason, we have a number of correction methods. Like we have BH, you have Holm correction, Benjamin-Hochberg correction. There is some test known as Utility or something test basically. Okay, and like that also we have more than 10 or 15 different methods basically. Okay, BH is very highly used, followed by Holm. Holm is the very highly stringent method. Really highly stringent methods. Doesn't matter which method you are making use of, like for now, because you are learning, but you should know that what is happening. Why are we making use of these cases? Why are we trying to adjust the p-values, right? So the p-value correction is simply done in order to take into account the multiple hypothesis correction, to take into account the multiple hypothesis problem. Why? Because the error propagates in case of simultaneous hypothesis testing. Okay, guys. So for that reason, we need to correct the p-values. And once we correct the p-values, the values are named as adjusted p-values, basically. Hope this has answered your query, basically, Priyanka. Right? So now the values has basically transformed, and these values are named as the adjusted p-values. So now let us see the result that we will get. We'll have how many values? See. So we'll have the genes as the column 1. We should have the log fold change. We will have the p-value. We'll have the adjusted p-value. And also we have one more statistic named as the B statistic, basically. This I'm not explaining you right now. Please go and read about this by yourself. Okay. So fine. So when you make use of a package named as lemma in R, basically, lemma package in R, so it gives you the result in this particular format. Also T statistic, it will give you T statistic. Okay. So you will have the gene A, you will have its corresponding log fold change value, you'll have the p-value, corrected p-value, and the corresponding B statistic and the corresponding T statistic here. And now what you will try to do, because you are interested to find out the genes which are significantly different. So what you can do, very simple. You can apply the filters upon the log fold change and the adjusted p-value, because you have corrected the p-values, right? So what you can do basically now, you will select the genes which are greater than a required log fold change and gene adjusted p-value less than your threshold. This threshold depends upon you. You can choose any threshold basically. Ideally, like normally the papers that you will read in bioinformatics and all, you will find the log fold change threshold as 1 and p adjusted p-value as 0.05 basically. Right, guys? So these are the two thresholds that we normally use. Okay. So by applying these filters, you will get the genes which are the genes that you are basically your differentially expressed, right? So now if you're going to plot them, so you will get something like this. So you'll get something like this basically. Okay. So you'll get a majority of the genes in this region, the genes which are not differentially expressed, and this value is 0 here. You will get like very less number of genes in the upper like region, positive, and here negative. Okay. And these are your minus log 10 p-values. Sorry, like here, where? Yeah, sorry. So here basically, it is log 10 basically. Like when we take the log of the p-value, it is on the 10 base scale basically. So it will become then 10. Okay, log 10, 10, it will become 10 base 10. So 1, so it will become 10 basically. Right. So this is how we get the results. But to select the genes, you will apply the filter upon the adjusted p-value. Plotting, you are making use of this p-values, negative log of p-values. But for selecting the genes as a differentially expressed, you will apply the filter upon the adjusted p-value, right? And these genes that you have selected are known as DEG or differentially expressed genes. Now the point is, what are you going to do with this set of the genes, right? So can anybody tell me what we do later basically with this set of the genes then? Anybody?
सर इसका पाथवे एनालिसिस कर सकते हैं ताकि ये पता चल सके कौन सा कॉमन पाथवे इफेक्ट होगा.
Good. So Hariom is definitely saying right basically that after that we basically go for the enrichment analysis basically. अभी हरिओम आप क्या कर रहे हो basically?
सर ग्रेजुएशन कर रहा हूँ. यूजी.
ANDC से ना?
यस सर.
Right. So then we perform the enrichment analysis, and which the target of which is to basically find the pathways, to find the set of the pathways in which the genes are enriched for. Okay, I suppose that you got total 10 genes, but hardly you will be able to understand the genes, right? You are more of interested to understand the genes are fine. Like what I'm going to do with the set of the genes. You will be more interested to find like are the genes involved in cell cycle, or the genes are related for the insulin regulation something, or the genes are regulated for responsible for which particular set of the pathways. That is your main task that you're interested in, right? So this is named the enrichment analysis or the pathway analysis. Also, we try to look at the genes for their, we try to look the genes for their PPI network, the protein-protein interaction network, that how the genes are going to look basically, right? So till here, if anybody have any question, you can ask me now.
So can you explain the filter part again?
Which part? Filter?
Yeah. Yes, sir.
Filtering is very simple. Filtering मैं दिखाऊंगा अभी आपको प्रॉपर करके तो समझ में आ जाएगा बेटर, ठीक है?
Okay, sir.
Fine. So is this screen visible, guys? Is the laptop screen visible? Please change basically, guys.
Yes, sir, it's visible.
ठीक है, fine. चलो मैं आपको ना डायरेक्टली पहले रिजल्ट दिखाता हूँ तो यू विल गेट इंटरेस्टेड लिटिल बिट। ओके, मेरे ऑलरेडी लैपटॉप में डेटा लोडेड है। फाइन, सी, so let me show you one result, basically how the results like data look like. See, okay, fine. So okay, so like this you can see here basically that there is a gene, there is a gene, there is corresponding log fold change, its corresponding p-value and its corresponding adjusted p-value basically, right? The adjusted p-value will be one also so many times, right? So the data result will look like this basically, and you will have number of basically what these genes. Like here we have 17,849 genes, right? So this is how the data will look like. But let me load one data set for you. So let me stop sharing the screen first for a minute. Fine. So hope the laptop screen is not visible now, right? Can anybody please confirm if the laptop screen is visible or not?
No, it's not.
Chalo, good. Good. Now the screen visible. Our screen, please confirm.
Yes, sir. Yes.
Chalo, good. So see guys, so right now I have loaded a file named as DEA. Okay, so let me show you how the file is going to look like. Great. So see here, like guys, so here we have the gene. So today class I'll be it's mainly for the interpretation part basically, right? So fine. See, so we have a gene. We have the genes here right now. So we have total 27,755 genes. Then we have their corresponding log fold change. We have their expression, average expression value. We have the corresponding T statistic that was calculated. Then the corresponding p-value, adjusted p-value that I've told you, explained to you just right now, and B statistic, right? So now I'll be showing you how you can plot this data basically, right? And then try to understand how to interpret the genes, right? So for that, you need to load a library named as ggplot2. Okay. And now we need to visualize it. Okay. So DEA, ggplot, DEA. So, and after that, we'll give AES, X equal to DEA dollar. On the x-axis, we'll represent our log fold change. Y-axis, we need to take the minus log 10 DEA dollar p-values. And then fine. And then we just need to plot here right now. Okay. Definitely you can make it very beautiful, like we're drawing some threshold lines and everything. I will show you that also. Okay. But for now, just give me some time. Now see here. So it will plot this graph most probably. Awesome. Are you able to look at the graph, guys? Hello.
Yes, sir.
Chalo, good. So now you can see here in this graph that majority of the gene is lying where basically, like between the range of like log fold change approx 0.5 से लेकर के -0.5 से लेकर positive 0.5 के बीच में, right? There are very less number of the genes which are crossing the threshold of -1 Okay, and positive direction में 1 के पार बहुत ही कम genes हैं. Okay, right. And then, so just a minute, fine. Yeah. And also then p-value. So now let us look at the y-axis, basically the negative log of p-values. So we know that like any gene which are greater than 0.05, we will select the genes as the significant at the adjusted p-value, basically, right? So then you will select the genes accordingly, right? So now what I can do basically now is that, see, so now I'm interested to expand history. Great. Now what I'll try to do basically is, see, I will try to create a filter here. So DEA dollar log fold change, and I will take the absolute of this as a 1. Okay, not 1, but we can take as the 0.8, suppose. And fine. So, and DA dollar adjusted p value less than 0.05. Okay. And then C, great. DA underscore filled. Dim DA underscore filled. See, 114 genes only. So view DA underscore filled. Now you can see here, this is the filter data, right? So we have only 114 genes, right? So now I will be interested, like, and how we filter these 114 genes? By passing the threshold of log fold change 0.8 and absolute, because we have the negative sign also, right? And the adjusted p-value should be less than 0.05. Now these are the genes, right? So you can see here that these are the genes that you have selected and got, basically. Right? So these are your genes of your interest. Okay. Now what we can do with these genes? Okay, so definitely like in the plot also you can see, highlight those genes. There are ways, but right now I'm not interested in all those things. Okay. I'm more of interested to like letting you know the complete pipeline, like what are the different steps which a biologist or bioinformatician is interested in, basically. Right. So now what we'll be interested to basically now further to perform the enrichment analysis and to select the pathways in which the genes are enriched for, right? So the genes are what? 114, right? So now I will load one more library, and this library is named as GeoProfiler, GProfiler 2, right? Great. And now GProfiler 2 and then Ghost. And I can put a question mark, and then it will give me the help function of this, basically. See here, it is loading, basically. So see here, guys. So now it is in the right hand side description section, you can see that it is given with the help function of this particular function, basically help of this particular function, ghost, GOST. So you can see that I need to pass the query. Query is the set of the genes which I am interested in and find out. Organism name, Homo sapiens, ordered query, multi-query. These are the different parameters. Significance, threshold that you need to pass here. Okay. Exclude IEA. I will have to look into this, basically. User threshold. Okay. So the threshold that you will pass, by default, it is 0.05. These are the correction methods. Okay. So there's a different correction methods basically like bound for any FDR, false discovery rate, whichever you want interested to provide basically. Custom BG, what is the background set of the genes that you will pass? Okay. And then all these things. So let me try to run this function first. So GOST. Okay. And now here for the query, I will pass my G list DEA underscore field dollar gene. Okay. Then custom BZ is equal to DEA dollar gene. So I am providing the complete set of the genes which I have in my data set. These will be used for the background. What is the meaning of background and all? I will try to explain you in 5 minutes last time basically. Okay. Then suppose correction method we provide here as, so suppose that bone ferroni. Okay. Then this particular like user threshold I'm providing at 0.05 only. Okay. Highlight, let us do, I've not used highlight equal to true sometime basically. Highlight equal to true. Chalo, and then अब इसको run कर देते हैं. By default organism is Homo sapiens, yes, fine. Res. So now it is doing what basically? You can see here that it is now running enrichment for these set of the genes by keeping into account or keeping the complete set of the genes which are present in the data set as the background. And now it has given us some results. So let us see. Fine. So it has given us something. So res dollar result I'm interested in. So result. Now I'll check my result basically. So there are total 134 rows basically, right? Fine. So let us now look at this. See. Fine. So fine. So these are the results that we have got, guys. And you can see here that the term name means the biological pathways. As you can see here that a response to external stimuli. Okay. Immune system process, immune response, response to other organism, external biotic. So there are different sources now here. Like you can put here, let me show it here to you, result, dollar, source. And you can see here that, and we are interested to find out the unique of them. And now you can see here that 1, 2, 3, 4, 5, 6, 7, 8. So there are total 8 different databases from which the pathways have been enriched for. For example, GOBP, gene ontology biological process, gene ontology circular compartments, molecular functions, HP is, I guess, human proteome, KEG, reactome, transcription factors, and wiki pathways. These are the databases which have been used for the calculation of the pathways basically. Now, suppose you are interested in the KEG ones. You want to look at the KEG ones basically. So very simple. Like for now, I can put my here also KEG. And now you can see here that these are the different pathways that we have got from the KEG. Tuberculosis, pertussis, nod-like receptor signaling pathways, leishmaniasis, KEG root term, tuberculosis, testicular differentiation, and all. So these are different term names or the biological pathways that you have captured and find out basically. Right. Now the point is again, suppose these are the results that we are looking at, we are able to see, right? There is also some particular parameter or the argument which we can pass, and it will then give us the set of the genes also, the genes which have been found responsible for this particular pathway. Okay. I can change that also, but right now forget about that. Now, we are also getting the corresponding p-value of this. Let me find that where. Yeah, here you can see. Now suppose that I'm more of interested to visualize this data. I'm interested to visualize this data. So how can I visualize that? Okay, so what we do, very simple. So fine. And now I'm interested to visualize only my BP, basically. Suppose Geo BPC here. There are total 82 entries. So there are total 82 pathways which we have got from the Geo BP. Suppose that I'm interested to visualize only 20 pathways. I'm not interested to visualize all the pathways, but only 20 pathways for now. Okay. So first, and these 20 pathways are the pathways having the least p-value. The lower the p-value, highly the significant the pathway is. So let me first filter that particular data here. Where is the source? Fine. And then you will provide here GeoBP result. So this is the task number 1, that you will filter BP terms, right? Now you can check here dim BP 82 pathways, right? Now I'm interested to basically sort them first. So how we can sort them? See, order dollar. Oh, sorry, order BP dollar p values. So now we are going to sort them based on the p values, basically. BP underscore sort. Now the data has got sorted, and I'm interested to select only the 20 first. So BP underscore sort. So I have selected the top ones, basically. Oh, it giving me error. Oh, sorry. I need to pass the upon the rows, basically, not the columns. Great. So BP underscore sort. So now you can see here that second. Sorry, it is top, basically, not sort. Fine. So now you can see here that we've selected only 20 pathways, right? Which are these 20 pathways? These are the pathways: external stimulus, immune system, other organism, biotic, and all. Now we want to visualize it. So how we can visualize that as well, right? Now see, so we'll draw a simple bar chart. How we can do that? So क्या करना चाहिए हमें? We need to plot it, right? So ggplot BP underscore top AES x equal to BP underscore top dollar. On the x-axis, I will represent my p-values, and even in the negative log of 10. Okay. Then for the y-axis, I will give the order of my pathways. Okay, so BP underscore top dollar pathways term ID. Great. And then I need to mention here geom underscore bar. Oof, it has given me some error. Fine. Here. Great. So oof, it has given me error again. So fine, like I'm getting the error. So what I can do, basically, I can directly copy paste my code. Geom underscore stat underscore bar something it will give, basically, I know. Okay, so most probably. But that is not a big problem. These are the technical things, right? So you do not need to worry about the technical things. I can simply put a line of code here, correct the code, and it will give me the corrected code. You will see that. It will figure out everything, like what are you interested to do, just by looking at the code here. Okay. Now you see that it's adjusting, trying to like make some adjustment to its own approach. Okay. Hello, hi Anjali. Hello. So Anjali, I'm just taking 5 more minutes and then you can start. Okay. So
Hi.
I'm really sorry for the delay, basically.
No, that's completely fine.
Fine.
Yeah,
So just give me 5 more minutes. Okay.
sure.
Fine, guys. So now you can see here that it is all given me the corrected version of the code. Okay, so I will do now very simple. I will simply किसको copy करें हम? So it is giving me all the different possibilities. So ये वाला copy करते हैं because ये वाला order वगैरह सब कर देगा. ठीक है? Awesome. चलो. Normally ऐसे नहीं उल्टा order करना चाहिए था इसको. इसने ऐसे कर दिया. That's fine. We can correct that. Not a big deal. Now you can see here that okay. So इसको सही कर लेते हैं. Term ID चाहिए हमको इसमें basically. Term name I am interested, right? Term name. Reorder. Okay. Great. So now you can see here basically that we have got the corresponding p-values upon the x-axis and the related name of the pathways upon the y-axis, right? And trust me, like you can do whatever you are interested in. Like definitely, like I can arrange the bars in the descending order. This bar will go top by simply like changing one small code in the like passing one argument in the code, and it will make the bar opposite direction. And that is what we are interested in visualizing basically, right? We do not visualize in this manner. These are visualized in the reverse order, right? This will be the top, this will be on the bottom, but that doesn't matter right now, right? See, so on the y-axis, we are renaming the pathways, and on the x-axis, we are giving the path, like p-values. What can you comment upon based upon this? So very simple. You simply say that the genes that you have found as a differentially expressed set of the genes out of the total genes, the pathway having the least p-value or most significant p-value is basically the response to external stimuli, right? And these are the different pathways that you have found. Okay. Now the point is, what are you going to do with this set of the genes further? Okay, the answer is very simple. Now you will be interested to basically validate these set of the genes in other data sets. So what you will do, you will simply download a number of data sets from other databases. You will perform the same process there also. And the genes that you will find from there, you will try to validate that what is the intersection of the genes that is being found from all the different databases. And then you try to make some machine learning based model with those set of the genes, right? And if you are getting some good validated results upon at least 10 to 20 different data sets, then you say that, yes, the set of the genes that you have got has the potential to be called as the signatures. And that is the real meaning of signature, basically. Because when we perform the differential expression analysis, the gene is already differentially expressed in the two conditions. It means what? It is highly expressed in A or B, whatever order, doesn't matter, right? So it is differentially expressed in one of the conditions. Then you have filtered the genes based upon your significance level and all. So you know that the genes are significant even. Then you validated those findings in at least 10 to 20 different data sets as well. And the genes, like suppose that you are left only with the 10 genes after performing in 10 different data sets. So now definitely like you have got a very less number of genes to validate. So that is a great result. Then you'll be interested to basically, then you will make some machine learning based model like logistic regression, random forest, SVM, okay, or some ANN based models. Okay, you can make some any model basically. And then you will be basically making the model to and report the prediction or the classification power of those genes in the two different classes. Okay, and then you will claim that the set of the genes that I have got has a AUC ROC score of 0.9, 0.8 or something. Okay, or specificity of my genes is this much. And so these are the genes having the potential to be called as the signatures in this particular condition. And that is the meaning of signature basically. Okay, and how do you find the signatures in the real world bioinformatics settings basically? Okay, so I will give you an assignment basically today here, and I will highly recommend all of you to try make full use of ChatGPT or any AI tool. Okay, the data set, download any data set of your interest from the Gina browser. Go and download any data set of your interest. Okay, provide the target variable name also to the dataset, then perform the differential expression analysis and the enrichment analysis and visualize your result in this particular format. Also make beautiful volcano plots. The plot that I was showing previously, okay, in which the genes are represented as the dots and the log4 genes versus the p-values is named as the volcano plot. So try to make these two plots and filter the genes. Trust me, you will be finding that you are able to do that. How? Make use of some AI, but you should be able to interpret the results. Okay, this is what I've tried to deliver and make you understand by the end of my talk for today. Okay, so this is all that I wanted to deliver for today, basically. Tomorrow, I'll be showing you the complete hands-on with the help of Mix Omics package. Okay, which is like right now, Anjali will be telling you in detail like what is Mix Omics or other basically like integration techniques and all. And I'll be showing you the hands-on of one particular case study example tomorrow using multi-omics. Okay, but and why tomorrow? Because now you'll be able to interpret your results. Now you'll be knowing what is the meaning of p-value, what is the meaning of the different pathways, what is the meaning of genes differentially expressed and all. So now you have a little bit good idea about these terminologies which are used in the world of bioinformatics. Right. So I'm open to questions basically now. If any of you have any question, feel free to ask me. I'm done on my hand. Thank you so much, guys, for joining. Is the background inclusive of the filter genes? Yes. Yes, Priyanka. Great. So anybody have any question, guys? So what all you asked to do in the assignment? Very simple, like just download any dataset of your interest from the Gina browser. Gina browser, the dataset that I showed you yesterday, database. And also download the metadata of the dataset and then perform the differential expression analysis. And after that, filter the genes and find the different enriched pathways and make an enrichment plot. Okay, Injusha? Yes, feel free to mute and ask question, like it will be good. Can you please provide a notes on our commands used today? You can just go and write on the chat or anything, like it will give you every code basically. That doesn't make any sense. Okay. Then what you,
Sir, will you
yeah,
share
Injusha,
the
बताओ.
recording? Will you share the recordings?
You are asking that will I share the recording, right?
Yes, sir.
So we will upload all the recordings upon our YouTube channel. So I will highly basically request all of you to please subscribe our channel today. Okay, so most probably within 3 to 4 next working days, we'll upload all the videos there. Okay.
Yeah, okay, sir.
Please channel subscribe कर देना लेकिन ठीक है. चलो good.
Yes, sir, sure.
ठीक है, thank you so much. What are the thresholds and FC value filters used generally? Because normally one as the fold change value and p value normally 0.05. Adjusted p value 0.05. Yeah. Any other question, guys? Thanks. Chalo, good. So then if none of you have any questions, then I'm done on my end. So thank you guys for joining. Now we will be having the session with Dr. Anjali. So hi Anjali, are you there?
Yes, Alok, I'm here only. Yeah.
Hi. Yeah. So guys, let me introduce Dr. Anjali basically. So she is one of my senior at IIT Delhi only. So she did her PhD in the field of computational biology with Dr. Gajendra Raghava, okay, who is one of the big shot in the world of bioinformatics. And currently she is working, and then she also did some industrial job. And currently she is basically the assistant professor at IGDTUW, which is Indira Gandhi Delhi Technical University for Women. And she's working in the field like computer science department, right, Anjali?
Yeah.
Right. But her PhD was in the field of computational biology. And
Yeah.
today we have Dr. Anjali, like she will be basically showing you the different directions upon the multi-omics different approaches and how they are used in the real world setting, basically. So over to you, Anjali. Thank you so much.
Yeah. Firstly, thank you so much, Alok, for reaching out to me for this wonderful session. And my name is Dr. Anjali, and also thank you so much for this wonderful introduction. My background is in computer science, and as Alok mentioned, my PhD is in computational biology. So, my today's topic will be on multi-omics data integration technologies and tools. I'll be keeping the session majorly theoretical because Alok already told me that you guys will be going through a lot of hands-on session before it, and it might be a little overwhelming for you people, and so it's better that I should go through the theoretical aspects about what are the technologies, methods, why this multi-omic integration is required in first place, all of these things. So feel free to ask me wherever you have any query, but this will be more generic, keeping the audience in my mind. I believe I have been told that it's a mix of bachelor's, master's, and PhD, right? Hello, I think.
Yeah, Anjali. Yes, yes, yes.
Okay, so I have kept my talk around that only, not keeping it too much complex around the things, and I'll be talking things in a very simpler way and using my presentation as just a prop to explain things. So let me quickly share my screen with you all. Alok, you have to allow me to share the screen.
Mute. I need to basically Anjali, अभी कोई request raise करोगे? I'm not getting the request here basically.
I actually did.
Share sharing option, एक second. Okay, host only, all participants who can share multiple. Yeah, now your screen is shared. नहीं, एक second, not shared. Yeah, your screen is shared now, Anjali. Hello, Anjali, आप mute पे हैं.
Now,
Yes, now you are audible.
okay.
And your screen is visible to me. Let me confirm. Guys, can anyone else confirm if the screen is visible to you also?
Yeah.
Yes, sir.
Chalo, good. So yeah,
So,
your screen is visible.
Alok, I can't see the people. I can just see my screen here. So if anyone have certain queries or they are posting in the, I need that help from you. If someone is just
Sure.
putting out their query, you can please let me know.
Great. So guys, please unmute and ask question then, okay? I highly recommend please unmute and ask questions.
So my entire screen is visible, correct?
Yep. Yes.
Okay. So this is a brief content or outline that we'll be covering in today's session. Basic introduction, various data sources, methods and approach, visualizations, and some important key points that one should be aware about while dealing with multi-omics data and while integrating that data to solve a particular problem. Fine. Now, you might everyone have already gone through these details, but this is just quick overview and let's say just quick summary of what you have already gone through. Alok told me that you guys are already well versed with what are the basic omics data types in one of your previous sessions. So just going through it, various omics data types include per se genomics, where you're dealing with like copy number assessments, mutation screenings, transcriptomic mainly around gene expressions profiling, microRNA expression profilings. Alongside every data type, there is you can see in bullets what are the basically technologies or techniques that are used to get that data, like protein proteomics. Proteomic profiling is like mass spectrometry. Then metabolomics, lipidomics, these are the different sort of omic data types at different levels within your body. So these are different, let's say at genetic level, some other information is more relevant, proteomic and all. So this is just brief overview of different omic data type. So what is multi-omic data type essentially? When you are mixing two or more omic data type to solve a particular problem of interest. And why is it important? So let's say one person at a genomic scale, all of us are very well aware about it that something which is there at the genomic level might not be very uh, evident or translated to the proteomic level. There are a lot of steps in between that comes from like particular gene level to the protein level, correct? So in that sense, every layer of information gives you more holistic, detailing information about what exactly is happening within the body. For example, here in the technologies part, let's say we have genomic transcriptomic data, and we have this copy number variations, structural variations. We can very well understand the functional effect of mutations. Okay, some mutation is happening, but how is it affecting the expression levels? That you can get from the transcriptomic differential expression data. That is how different linking across the, think it as a pyramid or as layers, where single omics is just uh, having relationships in just one single layers only, and there is no interconnection across the layers. Whereas in multi-omics, apart from within the layer connections, you have across the layers connections also, like linking what is happening at the copy number variation level with the differential expression level. Any doubts till now?
No, ma'am.
So, why this is important? This integration and interpretation helps you to get very reliable, fundamental, and detailed understanding of disease and their further clinical applications. For example, if I have to look at how, like histone modifications at the level of that, how essentially environmental factors are contributing to, silencing the genes or its activation and inactivation, I might be needing more than one omic data type to reliably conclude what exactly is happening within the system, correct? So that's where multi-omics data types are important. So these are some sources. It's not a very comprehensive list, but this is some overview of where multi-omic data you can get that to work with on different projects. TCGA is very uh, broadly used, very famous. The Cancer Genome Atlas, CCLE Cancer Cell Line, GTX also, it's mainly genome variations, transcriptomics, encode. Then you have variety of ATAC-seq, ChIP-seq, those kind of data sets for both human and the mouse cell lines. Then you have this blueprint IHAC data, which is DNA-seq, RNA-seq, ChIP-seq, different sort of data types. So this is some glimpse and overview of that. People are really working hard to generate this data, and there are a lot of data which are available even on geo as well at the different the genomic levels corresponds to variety of diseased or the normal conditions. Now, aim. I have briefly talked about it earlier as well. When we are talking about multi-omics data, it can help in variety of catering to variety of requirements, or help in variety of uh, applications. One, revealing the molecular signatures, like if I have to identify disease specific biomarkers that are very robust and reliable, I would like to see if that the same, let's say, certain gene is responsible for a certain protein, right? So if something which is happening at the genetic level is also evident and related at the proteomic level also, so that gives one such example may give you more reliability and confidence about your results. Now, identifying indicators of drug response. So there are data sets like links 1000, where across the different cell lines you will find the data about given a certain medicine or a drug, how the genomic profiles and expressions are changing across those lines. So let's say for a drug, I would like to see whether it is having some positive outcome on the patient health or not. So I have my genomic profile of a normal person, and for a diseased person or cell line where that drug has been given, whether it is able to successfully reverse the uh, expression profile or not. These sort of things helps you identify certain the indicators of drug response. This is one such example. Then construction of molecular networks. So one gene can correspond to multiple proteins and vice versa. There is one to many relationship, also many to many sort of relationships at different levels of biological entities, right? So how one thing is linked to another, miRNA, genes, proteins. Constructing those molecular networks gives you better understanding of disease mechanisms. And at what level? Some therapies you might see at the gene level, some might see at the miRNA level, mRNA level, something like that, right? So that this holistic pictures helps you to give better understanding about the disease mechanism, which in a way will help in designing better drugs. Now, again, prioritizing potential drug targets, like you can look at the similarity of two drugs structures, and their side effects profiles. So, like docking studies, how the similar they are behaving, how similarly they are binding to a certain target, those things. So, these are also helping in criteria for drug designing. Then, validating selected drug targets, guiding the validation experiments. So different sort of multi-omics data can also help in better designing the clinical trials also. Okay, what sort of patients you have to consider, what should be the right sample. That's why you see may have seen about that. Okay, for this particular drug, this age group, this is the efficacy. For example, cervical cancer vaccine, if you are taking it before a certain age, you may need three dosages of that, two dosages of that, and after a certain age you may need three dosages of that. So even while designing certain dosage experiments, many things, all of these data are very crucial for certain applications. So till now, we understood what is omics, multi-omics, and why is it required in first place. Now, of course, when we talk about different levels of data, even in our real life, there are certain challenges that comes with it. One, biological system complexity. Till date, there are many things which we are not even able to understand properly why this is happening. We may see it at the phenotypic level, oh, there these are exceptions, why this is happening. But biological system inherently is very complex and it keeps changing also. In the sense, not fundamentally, but your environmental factors or lifestyles, many things contribute to uh, certain changes. And then high biological variability across the samples also. So when we are designing a study, we also need to consider, that's why it's like what age group is considered, how much BMI is considered. Especially BMI is important when you are looking at diabetes or obesity related studies. Outliers, sometimes let's say when we are considering at age level, most of our samples, let's say, lie around 30 to 40 years of age, for example, and some around 80, 90. So outliers are from your mean average data, which is very far differently. Like here, if you can see, this red are outliers. These are very distant from the majority of data, very different from that. Then missing data. So when we are talking about like something which is there is a lot of missing data also available, can be available across the different omic levels. Why? Uh, let's say when we are talking about certain data, be it genomics or proteomics, getting one data as compared to other might be very price intensive as compared to others, and there might be challenges in terms of technological advancements which are there to get one data as compared to other. So for all samples you may not get all the data. So you also need to take care of when you are combining two different omics types, so whether it is available for all the samples or not, or whether it is available for, let's say, gene expression for all the genes, whether it is available for all the samples or not. So these are missing data and these also needs to be handled, and there are multiple ways one can do it. I'll briefly tell it. Here you might have read it already in one or the other session, like replacing with the mean values, or median, or totally discarding the samples if they are not that huge in numbers. These are some of the ways. Then data dimensionality means that you have very less number of samples, but then sometimes you have like thousands and lakhs of data points, like features. For example, genes, it's like 20,000, 30,000. You can't create some kit or a profile, let's say a diagnostic kit. 20,000 genes you can't use, right? 20,000 genes के pool पे you can't create a marker profile. Clinically, it will be a hell of a task to see those how they are changing across the uh, the samples or the people, right? So you need to reduce it to a certain meaningful levels where it is understandable and interpretable also at the clinical and the real level. And then again, low number of samples for certain disease conditions or for certain data types, it is difficult to get a lot of samples. That is also a huge problem while combining for multiple data integration, multi-omic data integration. Am I clear till now?
Yes, ma'am.
Thank you. So different approaches. We'll go through one by one. These are simple names as of now on my screen to tell you some, give you the glimpse of what exactly are the different approaches, can be supervised and unsupervised. Uh, I hope you are aware about supervised and unsupervised approaches. Or shall I brief it in one or two lines?
Please
Ma'am,
brief
it
it
would
in.
be better if you brief it.
Okay. So when we talk about as the English में as we are seeing it, supervised मतलब somebody have some information. Let's say if I'm seeing, let's say a machine learning algorithm would like to see uh, a cat, whether this is a cat or not, right? It's a simple algorithm. So they do have information of some samples already, whether this is a cat or not. In another way, like in biology terms, let's say if we have we need to identify, we need to build a model which can predict whether these are tumor patients or non-tumor patients, like samples, right? But we already have some information about uh, those patients. Let's say 100 patients, we already know कि, okay, these are cancer patients and these are non-cancer patients. Now using that prior information, you are building the model. Why is it important? Let's say genomics में there are uh, multiple genes, right? 20,000 or something we are talking about protein coding. Out of which, out of that pool, how many of genes are important to clearly predict the information कि, okay, these are cancerous, these are non-cancerous. For example, what information I need to see, oh, this is a cat. 4 legs, tail, usko, she should say meow or something, right? These are the features. How many of those features are relevant when I have to predict whether this is a cat or not? Understood? But I already know ki, okay, these are the things. This is a cat, this is not a cat. But I don't know which features are important. So I will design a model using X number of features to come up with which feature set or a combination of features is giving me a high performance in terms of identifying whether this is a tumor or non-tumor, this is a cat or non-cat species. Correct? Is that clear for supervised?
Yes.
Yes, sir.
So I do have labels, but I don't have information ki which is the right feature set, which gives me high performance. When I'm saying high performance, let's say मेरे पास sample था 100 का, 50 were cat, 50 were non-cat, 50 were tumor, non-tumor similarly. उनमें से कितना सही predict कर पाए उन features की मदद से. That will be the performance. Non-cat को non-cat और cat को cat. So that feature set which will give that higher number of right results is your right feature set to predict whether this is a cancerous or non-cancerous. और उसी के basis पे you design your diagnostic kit. One example, very brief and not in that depth. Unsupervised network के बेस के हिसाब से समझो, where you don't have any ground truth whether this is a cat, this is a non cat, but it's like clusters कि आपने सैंपल्स को बस डाल दिया सिस्टम में और किसी पैटर्न के हिसाब से यू आर ग्रुपिंग देम, लेटर ऑन यू आर आइडेंटिफाइंग कि ओके व्हाई दे हैव बीन ग्रुप्ड, की सिमिलैरिटी क्या है। करेक्ट। लेट्स से आपको एक पूल दे दे, आई एम गिविंग यू सम मिक्स ऑफ वो व्हाट वी से दैट कलर balls. Let's say there are red, green, and blue, and I'll just throw it. So you essentially would identify a certain pattern, right? Ki, okay, these all are blue color balls, these are green color balls. So there is some pattern जो ये algorithms identify करते हैं, some similarities, and they group them in clusters. Later on you elucidate that okay, what are the reasons behind why they are grouping together. Understood? So in one level you have certain labels, some ground truth. Unsupervised में you don't have that ground truth. ठीक है, फाइन, क्लियर,
यस मैम।
sure. And where is it important? Supervised का I gave the example, tumor and non-tumor का. Unsupervised is, for example, you already know कि एक drug is targeting particular gene or protein, right? That has been, वो उसके लिए clinical studies में it's been tested. We already have a string DB sort of databases where within our body how the different genes or proteins interact, वो network पूरा बना हुआ है. I have information कि this drug targets this particular gene. But these network based approach also help me to understand indirect targets also, which I can't elucidate directly from the clinical studies, right? अगर हम कोई स्टडी भी कर रहे हैं तो हम एक ही इम्पैक्ट देख रहे हैं, वी आर नॉट लुकिंग एट होलिस्टिक ओ हाउ एट एवरी चीन जो हमारी नॉकआउट स्टडीज भी होती है लाइक क्रिस्पर से या किसी से भी, दैट आर नॉट, they are looking at one entity within the whole system, right? Not giving the complete picture how these are linked. So these approaches are quite useful in that sense also, like disease subtyping. When I'm saying like tumor stages मुझे predict करना है कि what are the factors which are responsible for, or let's say there are three different clusters groups, let's say मैंने red green blue बोला, and those red green blue when we elucidated we identified okay these are certain stage के basis पे इन्होंने cluster कर दिया है. Correct. Now biomarker discovery also, as I have explained कि ये genes are important और better performance दे रहे हैं. These are response to treatments. Drugs का example जैसे पहले दिया था कि वो signature को reverse कर पाती है drug, हमारा जो पहले signature था, right? The signature, the normal reference signature for that particular age group, is it reversing back or not? One such example. Now different integration approaches can be supervised and unsupervised both. Supervised may we have mainly model based, concatenation based, and transformation based, whereas in unsupervised it's mainly around regression correlation, clustering, and network based integration. We'll go through it one by one. This supervised approach, this complete approach is talking about three different modalities. So machine learning model के बारे में जब I was explaining earlier, think about कि every different modeling जो टेक्निक है उस पे आप मशीन लर्निंग मॉडल लगा सकते हैं, चाहे वो क्लासिफिकेशन है वेयर यू आर प्रिडिक्टिंग लेबल्स येस और नो, या रिग्रेशन है जहाँ आप एक पर्टिकुलर वैल्यू प्रिडिक्ट कर रहे हो. फॉर एग्जाम्पल वैल्यू प्रिडिक्ट करना कि मुझे डायबिटीज के लिए ग्लूकोज लेवल प्रिडिक्ट करना है, तो कौन से फैक्टर्स हैं जो सही ग्लूकोज लेवल प्रिडिक्ट करके देंगे. तो उस केस में मेरा नंबर्स हो गए, नॉट कैटेगरीज या नॉट लेबल्स. फाइन. So concatenation based, model based, and transformation based. On the left hand side of the figure, you can see that these are like phenotypes, genomics, proteomics, metabolomics, different kind of data. व्हेन कंकैटिनेशन बेस्ड मेथड इज कि हमारे पास प्रोटीनोमिक एक्सप्रेशन है, जीनोमिक है, उसको हमने एक कंप्लीट जॉइंट मैट्रिक्स बना दिया कि एक तरफ हमारे सैंपल्स है, यहाँ तक प्रोटीन का है, यहाँ तक जीनोम का डेटा है, और कंबाइन पे हमने मॉडल बनाया, एनालाइज किया, इवेलुएट किया। दिस इज योर कंकैटिनेशन बेस्ड। मॉडल बेस्ड इज जहां आपने हर एक डेटा पे अलग अलग मॉडल बनाया कि अरे कौन से प्रोटीन्स इंपॉर्टेंट है, कौन से जीन्स इंपॉर्टेंट है, कौन से मेटाबोलाइट्स आर इंपॉर्टेंट, और बाद में उनको क्लब किया कि हाउ दीज आर लिंक्ड, पर सेपरेटली काम करके, ट्रेन करके। फाइन। ट्रांसफॉर्मेशन बेस्ड, हाउ मेनी ऑफ यू आर अवेयर अबाउट पीसीए काइंड ऑफ मॉडल्स, प्रिंसिपल कंपोनेंट एनालिसिस या ट्रांसफॉर्मेशन काइंड ऑफ मॉडल्स? एनीवन?
यस मैम, पीसीए के बारे में पढ़ा हुआ है।
ठीक है। So, when how many of you are aware about PCA?
मैम आई एम अवेयर।
Okay. Are there some people so that I can also just in one or two lines in simple terms I can tell?
Yes, ma'am. I am also aware.
Okay. So, when we are talking about transformation based, so there is a lot of data, correct? Let's say a lot of genes. Agar if I'm looking for heart disease, there can be XYZ many things. There can be your BP. There can be then sugar levels. There can be kidney functions. There can be a lot of things, right? So PCA is basically where you are simplifying in a way. It's a messy pile of certain features, and you are transforming into a more, let's say, less messy and easily understandable features. For example, if I have to see heart health, so heart health mein I can combine two features. For example, uh, let's say heart health ke liye I need my cholesterol profiles, sugar levels, BP, those things club karke I'm making it as a heart profile. This can be my PCA1, for example. Just vaguely I'm simple terms में explaining. Fine. तो वो I'm making it at every level of data, that transformations, and then working on the analysis on each individually transformed omic data, and then combining the results as concatenation based only. Fine. So these are the three different ways by which the data is integrated in supervised approach. Fine.
Yes,
Any
ma'am.
doubts till now? I assume no. Now we've talked about unsupervised. Unsupervised में भी again यही चीज़ें हैं कि मल्टी स्टेप लेवल पे you are checking it. For example, कि I am looking at network or how the clusters are forming at one level, उसके basis पे sequentially going to the other level. That is your sequential analysis or regression, association, correlation based. Correlation everyone understands, right? कि one is increasing or decreasing, how the other entity is behaving and parallel or in kind of relation to this, right? Is it clear?
यस मैम।
Okay. Clustering based is mainly around uh, where, as I've explained earlier also, you have a lot of data, you are making clusters and groups out of it based on certain factors. Like kernel based ke cases mein we can have some kernels or factors which are clustering the data. Bayesian clustering may be based on probability based. uh, Bayesian, like probability based, when I'm saying is that I know that there is a relation, but I'm also assigning, let's say in correlation, I'm saying ki, oh, zero point 0.9 is a how strongly they are correlated positively, right? But with what confidence also I can say this is zero point 0.9 strongly positively correlated, right? कि इसके 90% they are positively correlated, but उसका भी मेरा confidence क्या है? That is what Bayesian approach tells you. Fine. नेटवर्क में अगेन क्लस्टर आपको क्लस्टर्स देंगे बट कॉजुअलिटी नहीं देंगे. फॉर एग्जाम्पल जीन 1 और 2 how they are correlated, जीन और प्रोटीन how they are correlated, कि एक के बढ़ने से क्या कैंसर की ग्रेड बढ़ रही है कि नहीं एक्सप्रेशन से. बट नेटवर्क अप्रोच आपको देगा डायरेक्शन भी, कि कॉजुअल इम्पैक्ट क्या, इज इट जीन्स के बढ़ने से प्रोटीन्स बढ़े, बिकॉज इट इज अदर वे अराउंड ऑल्सो ना, कि प्रोटीन से फर्दर इट इज कंट्रोल, राइट. तो those things where network approach will help you. So that is how Bayesian clustering, Bayesian networks are different here. So this is a simple data integration tools which are there for across the different uh, I would say approaches that we discussed, like dimensionality reduction, which is correlation based, PCA factor analysis based. So जो हम PCA में बात कर रहे हैं ना, कि let's say PCA 1 2 3, you can think them as factors, कि किससे better explain हो रही हैं वो चीज़ें. Let's say if I'm saying heart health, that can be comprising of 2 or 3 features in itself. Fine. Kernel based again, कि कौन सा इंफॉर्मेशन है, कर्नल इज इम्पॉर्टेंट, जहाँ नॉन लीनियर रिलेशनशिप्स माइट बी लीनियर ऑल्सो, बट बायोलॉजी में इट्स लीनियर रिलेशंस आर हार्डली देयर, नॉन लीनियर रिलेशंस ही होते हैं, कि अगर कोई चीज़ 10 से 20 हो गई है तो ज़रूरी नहीं है सामने वाले पे 10 से 20 हुई है. इट कैन चेंज इन एनी मैनर, क्वाड्रेटिक और एनी अदर, राइट. नेटवर्क बेस्ड, एज आई हैव टोल्ड यू अर्लियर, वो रिग्रेशन भी हो सकता है और प्रोबेबिलिटी बेस्ड भी हो सकता है, कि आप कॉन्फिडेंस भी असाइन कर रहे हैं उनके साथ. देन ऑल्सो डीप लर्निंग बेस्ड. Deep learning is where you have really high dimensional data. You have some sort of ground truth also. In this figure, you can clearly see the color of the bars explains what basically that tool, their subjective importance or functionality they might be important in, like detecting disease association patterns. Every other tool or approach is important or playing a role in one or the other sort of uh, objective. फॉर एग्जाम्पल व्हेन आई वाज टॉकिंग अर्लियर, अगर मुझे ये देखना है विच म्यूटेशन इज असेंशियली इम्पॉर्टेंट, कि एक्सप्रेशन ठीक है चेंज हो रहा है, बट देयर कैन बी मल्टीपल म्यूटेशंस विच आर impacting that expression level. वो चीज़ आई कैन ओनली सी एट अ पर्टिकुलर a particular omic layer, और पर्टिकुलर मेथड माइट बी द राइट मेथड फॉर इट. फॉर एग्जाम्पल अगर मेरे पास कोई ग्राउंड ट्रूथ नहीं है, network based approach भी might be the right choice there. If I have certain ground truth, correlation ही सिर्फ मुझे देखना है, I'm not very interested in terms of any other details, then simple correlation based methods are important. Like SNF based method, if you can see here, network based approach, this is similarity network fusion. You have different networks for different omics layers, and then you try to see कि अगर इन सबको combine कर दिया जाए, how it will gonna look at a complete network level. So this is a simple integration. What are the data integration tools and methods? I will not, because of the time constraint as well, I will not go into the every detail of each and every method. However, I'll talk about one or two methods here, and all of the details, these papers which I have cited, they have all the details around these tools. You can go through them. They have beautifully also described their links, like MOFA, multi vector analysis. This is coded in R. You can go to their link, read their manual. It's pretty simple to understand how it functions, because basic level understanding is same across. There is some changes across the different tools and approaches, but essentially it's well written. Uh, Somebody where there are very nice tutorials within these tools and links. The people have put great amount of effort in documenting as well as explaining how they functions. So I will definitely encourage you to just go through this paper. I have given the name and the citation at the end. You can see that it's a guide to multi-omic data collection and integration. It's published in 2022 in computational structural biotech journal. Now, MOFA I have talked about, multi-omic factor analysis. Some of the methods I have skipped intentionally, because it would be an entire different session on one uh, class, let's say correlation or covariance based tools and methods, including their mathematical explanations, how they have been formed, their framework, everything. So this is just to give some glimpse about how it functions. When we're talking about feature and sample, essentially when we talk about essays, we can consider multiple different omic types, and every omic types have different features and samples. Samples remain same, but features obviously can vary. Genes, number of genes can be different, number of proteins can be different, right? We are translating it to certain features across versus factors. फैक्टर्स हो सकता है कि कुछ फीचर्स को कंबाइन करके कि अक्रॉस द सैम्पल्स मैंने ये डिसाइड किया कि ये सैम्पल को आई पुट इन दिस क्लास. फॉर एग्जांपल फ्लावर्स हैं कि ऐसे ऐसे फ्लावर्स को, ऐसे ऐसे सैम्पल्स को आई पुट इट इन दिस कि ओके रेड कलर फ्लावर्स, तो जितने भी रेड कलर के थे उनको एक में ग्रुप किया, उसको मैं एक फैक्टर बोल रही हूँ. फाइन, जैसे हेल्थ के केस में बोला था कि हार्ट हेल्थ, आई एम क्लबिंग दोज फीचर्स, सॉरी सैम्पल्स, और उनको एक फैक्टर्स की फॉर्म में एक्सप्लेनिंग. ठीक है. नाउ फैक्टर्स से जब हम सैम्पल्स की तरफ आते हैं, लेट मी जस्ट ट्राई एक्सप्लेनिंग इट इन अ वेरी फन एंड सिंपल वे. लेट्स से फीचर्स और सैम्पल्स की बात करते हैं, तो दिस वुड बी योर एनालॉजी कि हर एक के मेडिकल रिकॉर्ड्स समझ लेते हैं, विच आर क्वाइट मेसी, ओवरवेलमिंग. और फीचर फैक्टर्स की बात कर रहे हैं जहाँ सिम्टम्स के बेसिस पे, विच इज योर फीचर्स, हम उनको किसी हेल्थ थीम्स में डाल रहे हैं. ओके, दीज आर हाइपोग्लाइसेमिक और हाइपरग्लाइसेमिक, ये हाई डायबिटीज है, लो डायबिटीज है, हाई शुगर, लो शुगर, नॉर्मल, इस तरीके से. सो दीज आर फैक्टर्स. फैक्टर्स वर्सेस सैंपल्स अगेन इज अ थिंग कि अब हर एक पर्सन को वो कस्टमाइज थीम के अकॉर्डिंग कंडीशन एक्सप्लेन की जा रही है. ठीक है, फीचर्स को यूज करके फैक्टर्स डिफाइन किए गए, अब फैक्टर्स को यूज करके सैंपल्स डिफाइन किए जा रहे हैं. इज दिस क्लियर?
यस मैम.
ठीक है. So when I'm saying annotation of factors, see 1, 2, 3, 4, 5, 6, consider it as कि I'm saying कि tumor grades 1, 2, 3, 4 something like that. वो हर एक factor how it is, if I'm saying कि मैंने grade किसी feature के basis पे grade decide किया और मैं उस grade को 1 बोल रही हूँ, how it is varying across the different types. When I'm saying variance decomposition कि I'm saying कि ट्यूमर ग्रेड 1, और लेट्स से आई एम सेइंग हेल्थ प्रोफाइल में कि हाइपरग्लाइसेमिक, कि वो कितना वेरिएशन कर रहा है अक्रॉस द सैंपल्स, कितनी वेरिएंस एक्सप्लेन हो रही है. कि आई एम सेइंग कि कॉम्बिनेशन ऑफ फीचर्स से मेरा ये फैक्टर बना है और वो फैक्टर कलेक्टिवली कितना वेरी कर रहा है. वेरिएंस इज बेसिकली वेरिएबिलिटी कितनी है. जितनी ज़्यादा वेरिएबिलिटी है मतलब दिस इज समथिंग विच इज कॉजिंग वेरिएशंस इन अ पर्टिकुलर सेट ऑफ पेशेंट्स, राइट. तो वो मेरा इंपॉर्टेंट है, वहाँ वो चीज़ एक्सप्लेन करती है. एंड व्हेन आई वाज टॉकिंग अबाउट दिस फीचर सैंपल्स टू फैक्टर फीचर्स, एनोटेशन ऑफ फैक्टर्स, यू कैन कंसिडर कि वो जो हाइपरग्लाइसीमिक, लो शुगर, हाई शुगर, ट्यूमर ग्रेड्स, वो पाथवेज भी हो सकते हैं कि ये जीन एक्सप्रेशन सेंस साइकिल राइबोसोमल इनसे बिलोंग करते हैं, राइट. फीचर फैक्टर्स आई कैन ऑल्सो टॉक इन दिस सेंस. अब जब वो इंस्पेक्शन ऑफ फैक्टर्स की बात आई ना, these 1, 2, 3, let's say factor 1 or 2, you can see the small graph here, these blue and red dots, these essentially are your factor samples. कि क्या इन फैक्टर्स के बेसिस पे ये जो हमारे सैंपल्स हैं, these I can segregate, right? Clearly it is visible, correct? Am I clear? Consider it as PCA 1 और 2. जब आप PCA प्लॉट करते हो तो यू कैन सी ना कि क्या क्लस्टर सेपरेट हो रहे हैं कि नहीं हो रहे हैं, तो मीन्स दीज आर एक्सप्लेनिंग good level of variability across a sample group of samples versus another group of samples, right?
यस मैम.
So this is our multi-omic factor analysis, जिसमें input हमारा multiple data matrices है, आउटपुट विल बी वेट्स, सर्टेन वेट्स, फीचर्स, फैक्टर्स कन्वर्ट करने के लिए. की पॉइंट्स विल बी ऑफ कोर्स को अकरिंग सैम्पल्स कौन से हैं, अनरिलेटेड फीचर्स, जीरो वेट्स उनको देंगे विच आर रियली इनएक्टिव और कहीं कुछ रोल ही प्ले नहीं हो रहा है, मिसिंग वैल्यूज हैं. And then the downstream analysis where they'll help is like explaining the variance, gene set enrichment, which we talked about, annotation, and then sample visualization, how important these factors in segregating the samples, seeing any pattern within the samples, right? So this is multi-omic factor analysis. Now if we talk about this is essentially a Diablo method, which is data integration analysis, which this is here. If you can see, correlation covariance based, again PCA sort of things will also help here, where you have certain input data, you define relationships, how these data sets are connected in the sense, is it correlation metrics across the multiple samples. Then extracting latent variables are similar to what we understood factors पहले, like component 1 2 3 sort of things. अब उनमें से भी important features ही select करना. Every PCA, every factor might not be important. We might have like 20,000 initially features. Latent features consider principal component, हमारे पास हो सकता है 100 से 200 ही हो, उनमें से भी सब important नहीं होंगे. दैट इज योर सेलेक्टिंग की सिग्नल्स. उसको फिर आप मॉडल को फाइन ट्यून करते हो, ऑप्टिमाइज करते हो, एंड देन यू your output as biomarkers or relations networks, those sort of things. I will highly encourage you should go through this platform. Mixomics, Alok will be talking about tomorrow itself. This is a platform in itself having a lot of uh, different options to integrate multi-omic data. It's embedded there. Some coded in R, some have really good web plugins where you can have these are circos plots, these oval plots you can see here. These are your heat map correlation matrices. So all of these are there in this resource, which is offered in Mixomics Diablo method. So this is one such method. Another is similarity network fusion. So let's say there are different omics layers, A, B, C. You can see here. Consider it as like genomic, proteomic, metabolomic again, and then some other patient information within that. How different. So this is similarity network fusion for patients. अभी तक हम क्या करते आ रहे थे? How two genes are similar, or grouping of genes, proteins, how they are linked. But this is some different approach. I have also provided the links down there, where one talks about, let's say कि एक level पे how similar patients are. Let's say for here, based on this some let's suppose genomic data, these are 3 patient clusters which can be there. What essentially the feature is there which is responsible for across the clusters connections and within the clusters similarity across the patient profile. It's other way around कि हम genes और proteins को group करने की बजाय हमने वो information दी. जैसे I was saying कि group हो गया and what are the features which are important there. तो उस basis पे different data को use करके हमने patient clusters बनाए. हो सकता है इस केस में कोई पेशेंट 1 इस क्लस्टर में है, दूसरे डेटा को यूज करके दैट पेशेंट 1 माइट गो इनटू दिस क्लस्टर, राइट? डिफरेंट एक्सप्लेनेशंस कैन बी देयर, बट जब हम इनको फ्यूज करते हैं दैट विल गिव यू होलिस्टिक पिक्चर कि अलग अलग लेयर्स पे तो ऐसे हो रहा है, कंबाइन करने के बाद पेशेंट्स कैसे क्लस्टर कर रहे हैं. व्हाई इज इट इम्पॉर्टेंट? क्योंकि कुछ रिलेशनशिप्स may exist at just one level, not at the other level, which might not be of that much of importance because it may be translating. Or having a holistic picture always supports, right? It gives you deeper insights in a way, right? So here we can see कि हमारे पास patient subtypes आ गए हैं and how they are grouping you may later identify it. For example here कि हमने बाद में study किया कि groups किस information के basis पे हुए हैं. And one may be survival months कि one case में it's just 1 month survival. Here it's 48, here is 115. This is, you understood, right? पहले हम फीचर्स लेके चलते थे, हम क्लासिफिकेशन करते थे या कुछ प्रिडिक्ट करते थे. अभी हम ग्रुप कर रहे हैं क्योंकि हमारे पास ग्राउंड ट्रूथ नहीं है कि ये कैंसीरियस है या नॉन कैंसीरियस. पर क्लस्टर होने के बाद वी कैन सी कि व्हाट फैक्टर इज रिस्पॉन्सिबल फॉर कि ये एक ही क्लस्टर में इकट्ठे हुए हैं या ये सेपरेट क्लस्टर्स में है. फाइन. सो दिस इज सिमिलैरिटी नेटवर्क फ्यूजन. कुछ चैलेंजेस हैं क्योंकि ऑफ कोर्स मल्टी ओमिक डेटा कॉम्प्लेक्सिटी बहुत ज़्यादा है. फ्यूजन के टाइम पे इट्स अ ह्यूज प्रॉब्लम. ठीक है. और अलग अलग लेवल के फीचर्स को आई कैनॉट वेट इक्वली, राइट. सम माइट बी इम्पॉर्टेंट फॉर सर्टेन केसेस. फॉर एग्जाम्पल अगर मैं स्ट्रक्चरल डेटा की बात कर रही हूँ तो व्हेन आई एम डिजाइनिंग ड्रग्स दैट माइट बी मोर इम्पॉर्टेंट in terms of some other omic data, right? So setting up weights keeping in view of your problem statement will be more sensible. One way. There might be several other ways. This is one way. Now this is it's a some glimpse of what are the total some list of tools which we have already discussed. Their GitHub links, codes, and whether they can handle missing data, their APIs are available or not. For custom data means कि मुझे अपना कोई data देना हो तो कुछ डेटा दे ऑलरेडी बी हाउसिंग दैट डेटा ट्राई करने के लिए, समझने के लिए. कुछ डेटा कि मुझे वो डेटा अपना फीड करना है, कैन आई ट्राई इट आउट. देन दीज आर अदर टूल्स, इम्प्लीमेंटेशंस. नाउ कमिंग टू द वन ऑफ द लास्ट पार्ट विच इज योर विजुअलाइजेशन. कि ठीक है डेटा हो गया, एनालिसिस होगी, विजुअलाइज कैसे करेंगे इतने सारे डेटा को. वन वे इज सर्कस plots, which is circular map for omics data. Here you can see proteomics, mRNA, LNC, RNA, long non-coding RNAs, right? Antisense, similar that different type of data. Now, and you see these lines outside, these orange and blue lines, these are your expression levels for control. So this example is for IPF, idiopathic pulmonary fibrosis, one case study. उस केस में यू कैन सी कि अगर मुझे नॉन कोडिंग आरएनए को देखना है कि हाउ इट इज कोरिलेटेड विद प्रोटीन, आई कैन सी विद दिस लाइंस पॉजिटिव एंड नेगेटिव कोरिलेशन, एंड ऑल ऑफ दीज connections are above 0.9 correlation cutoff. See, you can see here that correlation also you can decide. There are multiple uh, tools which you can use, packages to draw this plot, right? So it's a very simplified, very interactive, intuitive, and I think it's really beautiful how you can uh, just see the links and relationships across the different omic data types. Now another one is also omicsnet. This is a tool which gives you like different level of layers. This is what I was talking about कि सिंगल omics में आपके पास अलग अलग layers के relations हैं, but when you're talking about different multi-omic, तो across the layers how things are related कि MIRNA is like how these are linked. So this is a wonderful. Again, opportunities or challenges बहुत हैं, we have discussed previously as well कि missing values है, noise है, poor biological interpretation है. अभी analysis तो कर लिया. These visualizations helps you do better understanding and interpretation of your results. That's why they are important. That is also a challenge. Distributed data storage, inconsistent sample annotation. अगर आप देखते हो a major challenge even with the TCGA data, sample IDs, how these are annotated. There can be different annotation libraries also. Some might be following something which is very old, something new. उनको कैसे combine करोगे? Right? Studies, all of these challenges exist. तो यहाँ opportunities भी है. You can design tools, you can come up with better methods, or you can come up with कि minimal information use करके कैसे experimental standards set किए जाए multi-omics data experiments के लिए. So all of these are still open research questions where one can really contribute. So this is around multimodal. To summarize, few things which are really important while you're dealing with multi-omic data is importance. Why every problem might not be requiring multi-omic integration analysis, right? So why you are doing it, the importance of it should be the first question. Similar to that, जैसे machine learning में भी बोलते हैं, अगर मेरा similar simple analysis है, I'm able to get my answer. Why would I invest in computationally intensive? Hello? Yeah. Alok, is there any question? This is the last slide. I can take up the questions then. And integration attempts are majorly best carried out while using the known biological knowledge. मुझे पता होना चाहिए. Sorry, am I audible?
Yeah,
Yes,
yes,
ma'am.
Anjali, you're audible.
Okay, so I'll just quickly go through that slide. That was anyway the last slide. Uh, Biologically, I should be able to understand ki SNP gene metabolite, what's the importance they are playing within the system, what sort of study will be benefited from what sort of data. तो इनहेरेंट बायोलॉजिकल नॉलेज होना बहुत ज़रूरी है, तभी इंटीग्रेशन अटेम्प्ट सक्सेसफुल है. सो दिस इज ऑल अबाउट द टुडेज टॉपिक. Am I still sharing my screen?
Yes.
Yeah.
Yeah. Now the screen sharing is off. Great.
I hope. Okay, any convention regarding handling? I did talk about it. You can have used different data, missing data imputation techniques. बहुत कॉमन है कि आप सारा सैंपल्स के अक्रॉस देखो. लेट्स से फॉर एक जीन के लिए आउट ऑफ 100 सैंपल्स 80 सैंपल्स में डेटा है, 20 में नहीं, तो आप उनकी मीन या मीडियन वैल्यू से रिप्लेस कर सकते हो. या फिर 2-3 में नहीं अवेलेबल है तो उन सैंपल्स को डिस्कार्ड भी कर सकते हो. कुछ कुछ ऐसी चीज़ें. सिंथेटिक डेटा यू कैन ऑल्सो जनरेट. मशीन लर्निंग में एल्गोरिदम्स हैं, स्मूथ एल्गोरिदम है, जिससे आप डिस्ट्रीब्यूशन देखते हो कि ये एक्सप्रेशन का डिस्ट्रीब्यूशन है, और वैसा ही सिंथेटिक डेटा भी जनरेट कर लेते हो अगर सैम्पल साइज कम है तो. व्हेन वी टॉक अबाउट लेस नंबर ऑफ सैम्पल्स, दीस आर सम वेज. अच्छा, व्हाट आर द थ्रेशोल्ड्स जनरली यूज्ड? लेट्स से अगर हम कोरिलेशन थ्रेशोल्ड्स की बात कर रहे हैं तो 0.9 इज अ ग्रेट कि 90% कोरिलेटेड है. बट अगर उसमें बहुत कम डेटा मिल रहा है तो आई थिंक 0.8 और मोर देन दैट इज ऑल्सो एक्सेप्टेबल. ये नहीं कि 50% ही है, वो तो फिर कॉइन के जैसे हो गया ना. 50-50 पॉसिबिलिटी वो बायोलॉजिकल सिस्टम्स में वैसे भी रहती है. सो एनी मशीन लर्निंग और प्रेडिक्शन मॉडल बाय डिफॉल्ट 60-50 वो वैसे भी दे देते हैं. तो दैट इज नॉट अ राइट अप्रोच, आई वुड से. There are any other, of course there are other visualizations also. These are few which I have uh, showcased. Network approach is also there. And then heat maps आते हैं different levels of correlations के लिए. IVG is also one tool. They talk about different chromatin level पे भी, इस पे भी there are different notations and plenty tools which does that. The papers which I have cited, I'll share this PPT if that's okay with everyone. Look,
Yeah, sure. Like if you are comfortable, then I have no issues.
yeah, it's okay because I have provided the links also. People can go through that. And those papers where I'm talking about visualizations and all, they've also talked about other different visualizations which are also there. The papers which I have listed. So it's up to us which we find the most feasible. No,
Great.
it's not entirely up to you. It's also up to the problem which I have told earlier कि किस तरीके की प्रॉब्लम आप टारगेट कर रहे हो. वेदर उसके लिए कौन सा अच्छा शुड बी मिनिमम सैम्पल साइज फॉर दिस एनालिसिस. हाँ, सैम्पल साइज भी प्रॉब्लम स्टेटमेंट को ध्यान में रखते हुए है. अगर मैं क्लिनिकल वैलिडेशन की बात करूँ किसी पर्टिकुलर सेट ऑफ प्रॉब्लम्स के लिए, जैसे इफ आई एम टॉकिंग अबाउट होल जीनोम सीक्वेंसिंग डेटा या फिर म्यूटेशन लेवल पे या फिर ड्रग दी गई है पेशेंट को, तो वो बहुत कम सैंपल होंगे मेरे पास. उस केस में अगर मेरे पास 20 सैंपल्स भी हैं, आई कैन अंडरस्टैंड समथिंग फ्रॉम देयर. डिजीज स्पेसिफिक भी है. कई केसेस में आई माई नॉट बी हैविंग दैट मच ऑफ डेटा. सो इट्स नॉट अ वेरी one answer fit all the
Scenarios.
scenarios. It's mainly your problem specific.
Great.
Any other questions? I hope the session was somewhat might be contributing some to your existing knowledge. Where is it? Any other suggestions that having some sort of, what would you think that this is something we would like to study in depth, or this might be helpful for us, or this is something which is you know uh, interests us more and we would like to uh, take up it in more detail. We can come up with the future tutorials or sessions. May not be in this conference considering the scope, but maybe some other
Right.
the
So,
future.
Anjali, like Priyanka has raised her hand. Priyanka, do you have any question?
Not particularly question as per se, like ma'am was saying, like if there's any suggestion or something. Like going through the session right now, there were a lot of possibilities of the applications, like ma'am mentioned, regarding biomarker discovery or molecular pathways or drug target discovery. Um, the since the method that is being employed is have some sort of similarity, it becomes difficult, like which methods to be adopted for, let's say, vaccine design or drug target identification versus biomarker identification. So if there could be some a little bit of guidance or some clarity on to like what would be the difference, like in these
Okay,
methods that are described.
okay, understood. It's basically around in what scenarios, what studies, which method is more uh, preferable as compared to the others, right?
Yes, ma'am.
Okay, that's definitely we can consider in the upcoming sessions. Uh, okay, yeah, sounds reasonable. Even with the machine learning algorithms, that's a major thing. What algorithm is more suitable for what problems? Yeah, definitely. Thank you so much for Frank. It's a nice suggestion, and I think it's very much needed as well. Any other suggestions? No.
Okay, great. Then thanks Anjali for like the this beautiful talk and very It was really very informative. Okay. So then, guys, if any of you have any questions, suggestions, feedback, or anything, feel free to unmute and tell. Okay. Otherwise, I would definitely request, like yesterday, that please, if nobody have any question or anything, I would request to please come over the camera. It would be really great, and we could have a group photograph. Okay, because I'll be posting through the LinkedIn day after tomorrow, basically when the complete workshop will end for the day 1, day 2, and day 3. Okay, so it's a humble request all of you, those who are sitting in a comfortable position and think that you can switch on your cameras, please do so. Okay, so that we can have a group photograph. And after that, we will say thank you so much. Okay. So please, guys, please switch your cameras if it is comfortable for you. Guys, please do so. I'm really waiting. Okay.
Some people might be getting into that comfortable situation so that they can turn on their cameras.
Hmm. Yeah, live में एक second पहले let me stop the recording.